Drug clearance through metabolism. Once a drug enters the body, the process of elimination begins. The three major routes involved are hepatic metabolism, elimination in bile, and elimination in urine. Together, these elimination processes cause the plasma concentration of a drug to decrease exponentially. That is, at any given time, a constant fraction of the drug present is eliminated in a unit of time. Most drugs are eliminated according to first-order kinetics, although some, such as aspirin in high doses, are eliminated according to zero-order or non-linear kinetics. Metabolism leads to products with increased polarity, which will allow the drug to be eliminated. Clearance, CL, estimates the amount of drug cleared from the body per unit of time. Total CL is a composite estimate reflecting all mechanisms of drug elimination and is calculated as CL equals 0.693 multiplied by VD and divided by T-half, where T-half is the drug's elimination half-life, VD is the apparent volume of distribution, and 0.693 is the natural log constant. Drug half-life is often used as a measure of drug clearance because, for many drugs, VD is a constant. Kinetics of metabolism. First-order kinetics. The metabolic transformation of drugs is catalyzed by enzymes, and most of the reactions obey Michaelis-Menten kinetics. The reaction velocity, which equals rate of drug metabolism, equals the maximum velocity, Vmax, multiplied by the free drug concentration, C, and divided by the Michaelis constant, Km, which is added to the free drug concentration, C. In most clinical situations, the concentration of the drug C is much less than the Michaelis constant, Km, and the Michaelis-Menten equation reduces to the reaction velocity, which equals rate of drug metabolism, equals Vmax multiplied by C and divided by Km. Michaelis constant, Km, is the concentration of the substrate at which half of the active sites of the enzyme are occupied by the substrate. Km is the concentration of substrate at which half of the Vmax is attained. That is, the rate of drug metabolism and elimination is directly proportional to the concentration of free drug, and the first-order kinetics are observed. First-order reaction is a chemical reaction that is entirely dependent on the concentration of only one reactant. In such reactions, if the concentration of the first-order reactant is doubled, then the reaction rate is also doubled. If the first-order reactant concentration is increased fivefold, it will be accompanied by a 500% increase in the reaction rate, and so on. This means that a constant fraction of drug is metabolized per unit of time. That is, with every half-life, the concentration reduces by 50%. First-order kinetics is sometimes referred to clinically as linear kinetics. Zero-order kinetics. With a few drugs, such as aspirin, ethanol, and phenytoin, the doses are very large. Therefore, C is much greater than Km, and the velocity equation becomes V, which equals rate of drug metabolism, equals Vmax multiplied by C and divided by C. So it equals Vmax. The enzyme is saturated by a high free drug concentration, and the rate of metabolism remains constant over time. This is called zero-order kinetics, sometimes referred to clinically as nonlinear kinetics. A constant amount of drug is metabolized per unit of time, and the rate of elimination is constant and does not depend on the drug concentration. Note the difference. For studying pharmacological science, go to the link in the description box below this video or in the comments section. Thanks for watching and happy studies.